I wish to introduce you next speaker. She comes from United States of America. She flew to our beautiful country and to this conference. Professor Dr. Jeanne Loring. She's a globally recognized pioneer in development of human pluripotent stem cell research in both industry and academia. She is recipient of Stem Cell Pioneer Award, Rosalind Franklin Society Award, was elected to American Institute for Medical and Biomedical Engineering, and was named one of the most influential women in advanced therapies. Thank you very much. So the floor is yours.
Thank you. Thank you very much, Martin. Thank you for inviting me. It's a real pleasure to be in Slovenia again. I was here when I just finished undergrad work, and it was pretty much, hitchhiking through. So I'm great. It's great to be able to spend more time this time. I'm going to tell you first about pluripotent. It's all about pluripotent stem cells. These are the cells that can divide forever and make any cell type in the body. I'm going to tell you three stories. The first is the clinical application that we've developed for using pluripotent stem cells. But I'm also going to tell you about our efforts to use these cells to help rescue endangered species and for understanding the effects of microgravity on the human brain. My disclosures, I'm the founder and stockholder and scientific advisor for Aspen Neuroscience. And Aspen is currently doing a clinical trial for autologous stem cell-derived neuron replacement therapy for Parkinson's disease. I'll tell you more about that. And I'm also a scientific advisor for a number of other companies and nonprofits, and I'm on some ethics boards. So pluripotent stem cells are unique. They only exist in cell cultures. These are not cells that you will find in your bodies. You did have these cells, but only 5 days after you were a fertilized egg. After that, they start going away because they're the cells that form you. So we can, amazingly, capture them in a cell culture, and we retain all the potential to make everything in the body. They are also the only non-cancer cells that never senesce. They will continue to divide forever, and there are some interesting consequences of that. So these are the two sources. In 1998, Jamie Thompson published his work on making embryonic stem cells from human embryos. These were embryos that were discarded from IVF procedures. And then in 2007, Shinya Yamanaka made a real breakthrough, which won him the Nobel Prize in 2012, in which he showed that you could take skin cells, ordinary skin cells, and epigenetically reprogram them so they would become exactly the same as embryonic stem cells. So this is going into the clinic. These cells derived from pluripotent stem cells are going into the clinic. And these are just some examples. There are now clinical studies for age-related macular degeneration, for retinopathies, for Parkinson's disease, several of them, epilepsy, leukodystrophies, cancers, graft versus host disease, heart disorders, and diabetes. These are all in clinical trials right now. So to focus on Parkinson's disease, it is, I'm sure you're familiar, it's a movement disorder in which people lose their control over their fine motor movements. They can freeze, they can become unbalanced, or they can have a tremor. And it is caused by the loss of a specific cell type in the brain. These are dopamine neurons that live in the substantia nigra part of the brain, which is pointed out there in the figure. And these cells, then the dopamine neurons in this part of the brain connect with the other part, this other part of the brain, and the striatum, and that's the connection that gets lost in Parkinson's disease. So people do not have any symptoms of Parkinson's disease until around half of their dopamine neurons are already dead. And then it comes on gradually, but it is degenerative, and there is currently no therapy that will stop the degeneration. So the reason it's a really good candidate for cell replacement therapy is that there's historical evidence. That's pretty rare. Back in the late 1990s and early 2000s, several hundred people were transplanted with little tiny bits of fetal tissue from about 6-week-old fetuses. They took the part of the brain that it was going to give rise to the substantia nigra, so they're dopamine neuron precursors, and transplanted them into, and this is important, into the site where the dopamine neurons connect. We didn't try to regenerate the substantia nigra, but rather put the cells directly in the striatum where they could make connections. So most of this, essentially this kind of work has died out because nobody had any improvement. There are a lot of reasons for that, which I think will become clear when I tell you about our approach. But a few of them had 20 or 30 years of drug-free existence. It's as if they never had Parkinson's disease. So that's where the hope came from. And the idea took flight rather rapidly. I want to show you how people are using cells to replace neurons in the brain of Parkinson's patients. So there are 4 different kinds, 4 different approaches. One is the allogeneic, which was mentioned earlier. That is, they're not matched. That means the cells come from a single individual, and they could be embryonic stem cells or induced pluripotent stem cells, Jeanne Loring cells. They are then made into dopamine neuron precursors and transplanted into the brain, the striatum, of the Parkinson's patient. Another approach is to try to match the major immunohistocompatibility antigens with HLA matched. This is a approach that has been taken in Japan, where it is possible to get a very close match because the population is not very diverse. The idea of using autologous cells was ours at Aspen Neuroscience. And in this case, we take cells from each individual, we make them into iPS cells, we make them into the dopamine neuron precursors, and then transplant them to patients. There is interest now, but nothing is going on yet, using hypoimmune cells. In this case, you knock out, using CRISPR, knock out the major histocompatibility antigens, so you create a sort of non-immunogenic cell type. And if you want to think about it a little bit, you might think of the pros and cons of that. And I'll tell you the major con, because the pros are pretty obvious. The major con is that your cells are always, your body is always being surveyed by the immune system. And cells that grow out of control, cancer cells, rely on these antigens to kill off the cells in your body when they start becoming tumorigenic. So that means that these cells will not have immune surveillance, and if they become cancerous, there's nothing to be done about it. Okay, so why use autologous cells? The obvious one is rejection, and an autologous strategy means there is no rejection of the cells. These are people's own cells. We don't have to immunosuppress them. Manufacturing is different, but not more difficult. In one case, you take one cell line and grow up billions of cells to be used for all your patients. And for autologous therapy, you do a scale out, which means that you develop techniques for doing a lot of patients in parallel. We don't have to grow a lot of cells. We only have to grow a few million cells for a treatment for these people. There is a question of genomic integrity. If you expand these cells, go on expanding these cells, that's a problem that has really, I think, probably caused a lot of companies millions of dollars because they didn't realize this. They get mutations as they divide. We have a lower probability of mutations, but still our release criteria, our quality control, does still look for those. But it's much less likely to happen. And then interestingly, if, for example, a person gets the first treatment and they don't get enough cells, the cells don't survive very well, you can't redose them if they don't belong to the patient. If they are the patient's own cells, you can redose them because the other cells will be instantly rejected. So I told you that pluripotent stem cells are unique, and I pointed out that they're the only non-cancer cells that don't senesce. So this is the darker side of pluripotent stem cells. their promise, of course, is an unlimited supply of cells from individual people, from me, from you. I have my own iPS cell line. I've used it for lots of things. And I have, most of my friends have their own iPS cell lines now. But there are hidden dangers in cell division. And this has been well known, but not really well recognized for quite a long time. This is the idea of bad luck cancer. This is a theory that Bert Vogelstein and his colleagues came up with some years ago. And it was based on really observational where cancers arise. They found that cancers were more frequent in populations of cells that divided. And they calculated that random errors that occur during DNA replication are a major contributing factor to cancer. That's why it's called bad luck, because there's nothing you can do about it. Your cells divide, they acquire mutations. And this bad luck component actually explains a lot more than environmental or hereditary factors. So here's some work that my lab did a very long time ago, 2011. We looked at nearly 200 cell lines, pluripotent stem cell lines, embryonic and iPS cells, and we discovered that there were repetitive mutations that occurred, really gross, large mutations. And if you look at the graph on the right, you'll see that every place where there is a red line means that chromosome was duplicated in a cell line, or that part of the chromosome was duplicated in the cell line. This happens over and over again in the same places in these cells. Okay? We know that's going to happen. So what do you do about it? Well, there's nothing you can do as far as culture conditions are concerned, except for treat the cells gently. There's evolution in the incubator. That means that as soon as you get one cell that has a mutation that gives it a selective advantage, it will become the dominant cell type in that population. And it wasn't until 2015 when we discovered that if you lost specifically p53, which is called the guardian of the genome, because it is mutated in 50% of cancers. We discovered that if the cells, if those cells, when those cells lost p53, they took over the culture again and again and again. And then a group at Harvard discovered that that p53 mutations, just by surveying all the banks of cells that people had grown up, even the cells that had been already GMP, that is, the cells could be used for therapy, he discovered that a lot of those had p53 mutations in them because nobody knew that they needed to look. So luckily, starting in around 2017, the FDA, the US FDA, and other regulatory agencies realized they were going to have to ask people to please look for this so that you aren't transplanting cells that are likely to become cancerous. Okay, so we can improve our luck. We can't fix it completely, but we can improve it by analyzing the cells at regular intervals. Avoiding excessive expansion, which we do with autologous therapy. We also, it's good to avoid gene editing because gene editing causes mutations, especially structural changes in the genome. And you have to look for them if you're going to do gene editing. There are many examples of structural aberrations, whole chromosomes being turned backwards and reattached, things like that, really gross changes that nobody noticed because they weren't looking for them. So you need to monitor the cell lines frequently. We use SNP genotyping, single nucleotide polymorphism genotyping. It's the same technique, in fact, exactly the same array that is used by 23andMe. And we do routine whole genome sequencing, comparing the cells as we go through the process. And we use a curated database. And what I mean by that is that when new mutations show up, they get added to this database. So it's current. So this was our solution at Aspen. We developed multiple levels of genomic analysis to ensure that the cells would be safe and efficacious. So this little circle here represents the procedure that we use. First, we ID the people using oral DNA. That's the identity that we will trace all the way through the process. because we don't want to give the wrong cells to the wrong person. We then harvest their fibroblasts and we sequence their fibroblasts to find out what intrinsic mutations they have. If they have intrinsic mutations that aren't dangerous, and they probably don't because they live to be 70 years old without dying, so we just want to make sure they don't have any mutations that are associated with Parkinson's disease, causal of Parkinson's disease, because we're not really sure that we can treat that yet. So then those cells are reprogrammed, and then these assays are done again, including another assay called Pluripotent, which is a method that we developed for making sure the cells were, in fact, pluripotent. And just to outline it really, there are two of these assays that we use, NuriTest and Pluripotent. And they are gene expression profiling tests. We use machine learning and bioinformatics to create a gene expression profile that predicts the fate of those cells. So for a pluripotent cell, it predicts that they will make all three of the germ layers. They'll make different cell types. For the NuriTest, it predicts that the cells will become dopamine neurons. And that's our release criterion, our quality control criterion. So this illustrates those two assays. This one, you can see there's this timeline of embryonic development starting with pluripotent cells when you're a blastocyst. And then there's a stage called specified, determined, and then differentiated. These are general stages that cells go through as they develop. And so we developed this assay based on a very large reference database that we created of gene expression profiling, of profiles of cells that we then asked whether they would in fact make three germ layers. And that was done by either doing an in vivo test in mice or doing an in vitro test, which is called an embryoid body test. We showed that the cells can make all three germ layers. So using those data and the algorithm that we developed, we started a web-based application at pluritest.org, and it has now become the major method by which people say that their cells are pluripotent. Thermo Fisher has licensed it, and they use it. They will do it for a price, and they're running, as far as I know, they're running at least 10 to 15,000 assays every year. We did the same thing for the neurons. In this case, we did their gene expression profile at the stage which is in orange there, which we guessed was determined. And the reason we had to guess was because we didn't know. It's a transient state. And so we needed to find out what gene expression signature predicted differentiation into dopamine neurons. And that's how we developed NuriTest. Okay, so we had developed all these assays. We had done animal studies to show that the cells were efficacious in animal models. We'd done preliminary safety studies to make sure that the cells didn't make tumors in the animals. And then we were ready to start a company. So the history of the company was that at Scripps, when I was at Scripps Research Institute, a group of people with Parkinson's disease and their advocates climbed Mount Kilimanjaro in order to raise $200,000 to start my project in my lab. And the important thing about that was that I could not get money from a grant to do this work because it was completely new. I didn't have any publications on it. It was really an experiment. And so they have done other things, and eventually this group, the foundation, run by Jennifer Robb, is the Summit for Stem Cell Foundation, raised $3 million over the next 6 years. And that was sufficient for us to be able to start getting grant money and to start a company. We started the company in 2018. I started with a colleague and my postdoc, Andres Bratlia. And then only 5 years later, Aspen announced that they had FDA clearance for their first multi-center, phase 1/2A trial for autologous dopamine neuron replacement therapy. So it was just a year later that they reported that the first person had been treated under this protocol. And then this year, they've announced the completion of their low-dose and high-dose cohorts. And in fact, this has not been announced by Aspen yet, but it was announced in a talk last week by one of the board members, So I can say that all 8 of the patients who were treated in that phase 1 trial are all doing really well. And these are the criteria. This is the preliminary work, and I think the most important thing here is there's a neurology report, and there's also a personal report, people's living standards. And we found that there's an average of 45% improvement in the clinical score, 71% in a secondary, more lifestyle clinical score. They have on and off time because this drug, L-DOPA, is metabolized, and so they have times in which their symptoms are full-blown and other times when they feel better. And they have on average 2 hours less of that and 1.5 hours of better time. So, my stem cell haiku for the future of therapy for Parkinson's disease is neurons bloom anew. FDA's blessing ignites hope. Parkinson's may fade. I found that ChatGPT is much better at writing poetry than I am. And so I've taken advantage of that, and it's really quite remarkable. I'm going to tell you now about stem cells in space. Is everybody ready? Okay. All right. There are other things you can do with stem cells besides treat people or even cure them, which would be marvelous. But we can also use them for some pretty far-ranging experiments that are completely different from what people are doing now. My stem cell in space project is funded by NASA and also by the National Stem Cell Foundation. And this cartoon, I think, really says it well. I added the original cartoon says that it is the stem cells are told that they can be anything they want to be when they grow up. And in this case, the parental advice and the stem cell says, I want to be an astronaut. Stem cells in space affect a microgravity on iPS cell-derived neurons. Okay, so you might ask why, but I'm not going to answer that. I'll tell you about it later. But the idea was because there are humans in space, there are humans for probably long-range space. The NASA decided they needed to start doing biological studies, and not with flies or mice, but with humans. And this, you can't really measure things in the brain of astronauts while they're up there. And so we came up with a sort of a surrogate for a brain of an astronaut by taking taking iPS cells from different individuals, a group of individuals, and we made them into the type of neurons that are represented in 2 parts of the brain so far. In our further studies, they will be representing 3 parts of the brain, the parts of the brain that are affected by Parkinson's disease, by multiple sclerosis, and by Alzheimer's disease. So we do the same thing. We take iPS cells, we turn them into neural precursors. In this case, they're exactly the same cells, for the dopamine neurons, exactly the same cells that are being put into people's brains. And then we take microglia, which are one of the immune system of the brain, one of the cell types is the immune system of the brain, and mix those together so that we can capture neurons plus immune cells and make organoids out of them. The organoids are really small. They're about, for dopamine neurons, they're about 200 microns in diameter. For the cortical ones, about 500 microns in diameter. And that's really important because if you try, we're going to send it up to space for 30 days. It's really important that the cells inside the organoids get nutrients by just diffusion because there's no blood vessels. That's how they're not brain. So we put them into little vials. These are what you might know as cryo vials, but we're not cryoing anything. They seal up really well. And so we put them in 1 ml of medium in a cryo vial, close it up, and then we send them to space. This is my haiku for neurons in space. Cosmic voyage starts. Neurons defy gravity. Life blooms in space. Space's embrace. These badges around the bottom represent the mission patches for the 6 missions that we've done. That's one of the cool things that NASA does. And this is what we do to get the neurons into space. We make the organoids in the lab, and then we take them at low temperature to Cape Canaveral in Florida, And there we have a lab. We assemble we put the cells into a holder, essentially, and then we turn them over to NASA. They put them in the Dragon capsule. And the Dragon capsule, of course, is on a Falcon rocket, and the Falcon rocket takes off. So the next day, day or two, they dock at the ISS. They get plugged into the incubator there. All they have to do is stay warm. And then 30 days later, they come down with the Dragon capsule, and then they're removed and shipped back to us. We start analyzing them then. Why 30 days? That was the right rhythm for astronauts going up to the space station and for supply missions, taking things up to the space station. So we published our work last year. It's called The Effects of Microgravity on Human iPS Cell-Derived Neural Organoids on the International Space Station. And this is, essentially, captures some of our results. That shows in the upper left, you can see the organoids. And the first thing that we discovered when the cells came back was that they were alive, because we didn't know whether they'd survive or not. We put them into culture dishes, and they started sending neurites out like crazy. So they were clearly alive and healthy and neurons, which was fabulous. And so we found, you probably can't see that, but there's a tiny little dot at the end of the arrow there on the lower right-hand corner. That's an organoid in one of the tubes. So one of the things that we discovered, I don't know if it's going to work or not. Yes, good. One of the things we discovered that was during the time they were in space, the dopamine neurons formed a network of synapses with each other, and they started bursting in synchrony. This is calcium imaging, and you just see them light up all together, all the neurons together, which is exactly what they do in a brain. They didn't do it before they left, but they did it while they were in space. And we have lots of data now that shows that the cells are electrically active. And we also found out, as I said, I think I told you in the first slide, that the cells mature faster in space. So we do a parallel experiment on the ground. We keep the organoids on the ground so we can have a direct comparison. And we do a comparison, and we discover that the markers of maturity are higher in the organoids that were in space than they were on the ground. It's probably the reason. I have a hypothesis that I haven't tested yet, but I think it has to do, if you want to think about this, there is no convection in space, which means that there aren't things moving around in those little vials. So that means that each one of the organoids, whatever it secretes, is going to stay right next to it. And so I think it creates a different environment, which leads to greater, faster maturation. And they also stop dividing. When we make them, they're still dividing. They stop dividing. And they divide less in space than they do on the ground. Now, I have to point out, this is not aging. This is maturity. These are still like younger than fetal cells. I want to acknowledge the people involved because it was quite a large team. There's a group in New York that collaborated with us in San Diego. And the main people that I was associated with are Paula Grisanti, who is the CEO of National Stem Cell Foundation, Jeanne Stoudemire, and me. And you can see in that middle picture that's me in the mask there. I actually got to do some lab work, which has not happened in 20 years. And one of the very cool things about this is that if you look on the middle of the top, you'll see that the vial is labeled Jeanne Loring. And that's because my own iPS cells went into space. Haiku. Tiny brains take flight, spinning thoughts in weightless dark. Learning in the void. Okay, last part. This is using induced pluripotent stem cells to try to rescue endangered species. Again, the cartoon. In this case, they want to rescue endangered species. And the reason this project started, was a field trip. I just moved to Scripps, and I had a pretty large lab. I wanted to do something nice for them because they'd been moving stuff across the street for a couple of weeks. So I took them to the safari park. It's the San Diego Zoo Safari Park. And it's 28 miles away. It's not that hard to get to from our lab. And we took a safari. And the safari was organized by the person who was in charge of something called the Frozen Zoo. His name's Oliver Ryder. And for the last, I think, my goodness, about 45 years, he has been collecting fibroblasts from lots and lots of different animals and freezing them. That's why it's called the Frozen Zoo. It was founded in 1975. Our Center for Regenerative Medicine was founded in 2007. This is the safari park. And on the right, you see a younger me and also Inbar, Frederick Ben Nunn, who was the one who took on the project. As soon as we got back to the lab after the safari, she said, I want to try to make iPS cells from fibroblasts from the Frozen Zoo. And she did. We published the paper in 2011. We had the cover in Nature Methods, which is a little bit rather dramatic. But we made the iPS cells the same way we made iPS cells from humans. We took the fibroblasts, in this case, they're ones that were frozen. We thawed them, we reprogrammed them, and luckily these two animals, the northern white rhino and the drill, were both able to be reprogrammed with the human factors, the human reprogramming factors. Now, there are only two northern white rhinos left. That's on the right is Najin and Fatu. They live in a protected place in Kenya. They are just about extinct. They will be extinct within my lifetime. And this on the left is Nola. She was the last of the northern white rhinos who lived at the San Diego Zoo Safari Park. And I was lucky to get to meet her before she died. We've gone on and now made from the Frozen Zoo iPS cells from 9 different animals, 9 different northern white rhinos and a couple of a closely related species called southern white rhino. We wanted to do that because eventually what we really want to do is to make eggs and sperm out of the iPS cells and make new animals by mating them in a dish, IVF. And I think this is really, this always seems remarkable to me. This is Marissa Carodi. She is the one who trained in my lab and then started the lab at the zoo. These are iPS cells from a northern white rhino, Angelifu, who died in 2014. Their iPS cells have been differentiated into beating heart cells. You can see them beat in the dish. This is, it's alive. However, we have also asked the question, what happens when these cells are allowed to divide? And the answer is the same thing that happens with humans. We just published this year the first reference genome map of the northern white rhinoceros. Now, the human was mapped many years ago at great expense, but we had to start all over. There was no information that allowed us to, we didn't have any background at all. So we were starting like the first people who were mapping the human genome did. And we discovered they have 82 chromosomes, which is kind of a weird distribution, but I find that now that I've learned a lot more about animals in the zoo, they have all sorts of different numbers of chromosomes. And we discovered that in this particular induced pluripotent stem cell line, there was a deletion, and it's marked by that little blue thing on chromosome 5. So this cell line would not be used to make gametes because we have no idea whether any of the genes in that region are important to be, to have 2 copies of. But there are lots of others. So to acknowledge the people who do this was a really remarkable collaboration. This was people from the zoo and people from my lab, and nothing like that had ever happened before. So I think the collaboration is apt. Together we thrive, ideas blend, dreams take flight, collaboration. And the major people involved in this are my former postdoc, Franz Josef Müller, who is now the head of the hospital at Kiel University in Germany, and all my lab members and all these people at the zoo. In the middle, you see Oliver, Ryder, and me last year. And 2011 was the first time anybody ever made a iPS cell line from an endangered species. And then in 2023, we had the first conference on making iPS cells from endangered species. And these are people from all over the world who are doing it with different species. So it has become, it's now a field. People are doing this. It's for biobanking. And I think it's a worthy effort. Not everybody agrees with me, but I think it's really important because what we discovered with the frozen zoo, that we can essentially, maybe get an animal back, then we might be able to get a lot of animals back because they are, in fact, becoming extinct at an increasing rate. So the haiku for the future of the northern white rhino is: gentle giants roam, stem cells bring hope for their lives, rhinos saved, reborn. Let's hope. The future of stem cells. Stem cells hold the key to healing and longevity. A bright future lies ahead. ChatGPT. And these are the things I've done with stem cells. And of course, the question is, what will other people be doing with stem cells? Thanks. Yes.
I have a question about your first part. Mm-hmm. When you treated the patients with autologous stem cells,
yes.
Or actually iPS from their fibroblasts.
Yes.
Is it right?
But we differentiated them into precursors before we transplant them.
Okay.
Nobody transplants
iPS.
undifferentiated cells. That would be crazy.
No. Of course, you transfer them to iPS and then you
into neurons.
Differentiate them back. So
yes.
2 times. And then when you said that this, you made genetic analysis, if they have some already Parkinson's genes or cancer genes?
We wanted to exclude people who had hereditary forms of Parkinson's disease in this first trial.
Uh-huh.
I don't think there's any reason why we can't treat people with their own cells, because remember, they don't start losing their cells probably till they're in their 50s. And so that means if we can restore the cells, we don't expect them to die any quicker.
Yes, I understand. But these are stem cells from old patients, not from young patients.
It makes no difference.
But there are epigenetic changes, perhaps. That
no,
you
there
don't
aren't.
detect,
I mean, that's the really amazing thing. We've done all the. We've even done the epigenetic test that was, I believe, it's Altos developed. It's methylation, DNA methylation.
Yeah, methylations.
We've discovered that the age of the cells from an old person, iPS cells, the neurons we make, is virtually 0. So they have the methylation profile of a newborn. Yeah.
Same. Which is, of course, otherwise would be dangerous for cancer.
No, I agree. But the cells, we're taking cells that were normally senesce, because fibroblasts don't divide forever, and we're epigenetically remodeling them. Oh. And they are reborn. They're
That's
young
amazing,
again.
Yeah. Well, thank you.
Okay, thank you. One more question here.
Yeah, even 2. The first one is kind of a very general question, but I never understood how does one totipotent stem cell knows when they stop to be a totipotent stem cell.
We tell them. So
okay,
we don't leave it to chance. We tell them. They don't know. They honestly have no idea what they're going to be.
But seriously, okay, a totipotent stem cell, they will divide. At one moment, it will stop. How does it know it? Okay, that's my first question. And second question, totally something else, is are you working in some way with George Church teams and so on?
No, George and I have different opinions about how to revive endangered species, so no. No, I mean, I like him very much. He's very smart, and a lot of things he's done are very much like things I've done, but we disagree about, I don't want to bring back a woolly mammoth, and I'm not interested in dire wolf lookalikes, which is the other thing that's been going on. I really want to bring back animals that are still alive, or bring them allow them to continue to live. So the question you're asking is really a profound one. We know how to direct them to become dopamine neuron or other neuroblasts, but we don't know all the mechanisms that occur within the cells in order to get them there. This is all based on embryology and observations of what their surroundings are like, what growth factors or lack of growth factors. Interestingly, the way we start them off to make neurons is to tell them not to be not to be mesoderm or endoderm. Instead of just telling them to be ectoderm, we tell them what not to do. And sometimes you have to do it both ways. I mean, it's really, I can't take credit for this. A lot of this work has been done by many scientists over many years. A lot with mouse ES cells, which were, have been around since the 80s. So yeah, the methods are for dopamine neurons, the methods are really very, very good. And we get precisely the cell we want.
Sorry, okay. I know you are fighting with the time, but I cannot help. I like crazy people. You are so sweet.
Thank you.
Crazy.
Thank you. Thank you.
This space stuff. Oh God, my pulse went up 30%. I really like. Maybe Elon Musk would like you.
Yeah, that's a good question. I actually would like to see whether he wants to send his cells up to space. A few $1 million.
So just one thing. When you implant the cells, how do you do it?
Oh yeah. So I didn't go into that in detail, but we have a new way to do it as well. We have an incredible neurosurgeon. We drill little holes in the back of the head, and then there's a very thin catheter that is placed into the striatum. It's actually the putamen, which runs this way. And so you drop off the cells as you pull the catheter out. I just watched the surgery for the first time. It was the most remarkable thing I've ever seen. And it's all done with MRI. So there's imaging and then imaging and then imaging to make sure they're on the right track. So they put them in the right place. But it was, to me, it's just as amazing as turning the cells into dopamine neurons in the first place. Yeah.
Okay, thank you. Thank you very much. Thank you very much, Professor Loring.